We go over the regulation of gene expression. Well, before we talk about the regulation, we need to understand what a gene is, and after understanding what a gene is, and then we understand what gene expression is. Now, a gene is a segment of a chromosome. So a gene is a segment of a chromosome that codes for a polypeptide chain, or that codes for a protein, or let's just say that codes for a trait. So that is what we call as a gene. So it's simply a segment. Now when we're talking about a chromosome, we know that DNA exists as a double helix. Alright, so DNA exists as a double helix. Now the way we're going to find this DNA is going to be so much, because it is so small, we can't even see that it is a double helix. And so you just see it as a line. The same way it is, you just see it as a line because it is too small. And so this is going to condense, it is going to contract and become so small, and then you are going to see it in this form, where we've got one like that, and then another one like that. So it condenses, and this is what we call a chromosome. So a chromosome is simply this DNA that has now condensed, and then one part has come joined with another part on the very middle there. So now if we can look at this, it has got different parts. This is a chromosome. So let me also write it as, to show it much bigger, I show it straight away like this. So say this is a chromosome, where this one is the one we can see there, and then this other one is the one we can see there. So there are some portions. So it has got segments. So because we know that DNA carries nucleotides, there are some segments. This segment, so only this one alone is called an allele. This one alone is called an allele. So a gene is a segment on a chromosome that is responsible for giving a certain trait. So for example, this portion I've put here can be talking about eye color. Well, this one is called an allele alone. It might be it is talking about eye color, but let's say it is saying the color of the eye should be blue. This is also a gene, I mean an allele, which is also talking about eye color. But this other one might say, no, the eye color should be black. So these two alleles put together form what is called a gene. And so for you to see which color you are going to have, the one which is dominant between the black and the blue is what you are going to have. So if black is dominant, you are going to have black eye color. If blue is dominant, you are going to have blue eye color. So that is what we call as a gene. Now, gene expression is a process by which information from a gene is used to synthesize a functional gene product. So this information which is in a gene, so this gene here is telling us of eye color. Now, we are using it to produce a functional gene product. Gene product can either be a protein or mRNA. Because this is DNA, DNA can be converted into mRNA. Okay. So it can be converted into mRNA. And at this time, mRNA is called a gene product. Or actually, DNA can be converted into mRNA, then mRNA into proteins. So same, proteins here are also gene products. So that is what we refer to as gene expression, the process by which information in a gene is used in the synthesis of a functional gene product that enables it to produce the end product. Well, after understanding this, we know what gene expression is. Now, what of regulation of gene expression? So regulation of gene expression is simply controlling how you form gene products That is to regulate. You are controlling them. How fast is the process? How slow is the process? So that is how it happens. Well, we can also say gene expression is a process by which information which is found on a gene is used to make either a RNA molecule that codes for a protein or a non-coding RNA molecule that serves other functions. Now, we know that during the process of transcription, here DNA has to be converted into mRNA. Now, the mRNA that is formed has got what is called introns and exons. Alright, so let's see. So this is the mRNA that has been produced. So it has got, let's say the ones in orange are introns, the ones in green are exons. So we have got introns and exons. Now, some of these have to be removed, because when we are talking about a formation of a functional mRNA, some of these are not functional, they are not necessary. So exons, these are the DNA sequences that encode for a polypeptides. So exons are the ones which give us a polypeptide. Alright. Whilst introns gives us what? Introns don't code for any polypeptide. So introns have to be removed because they will not give us any protein. And so the yellow ones are going to be removed during the process of splicing, exon splicing, and then the green ones are going to remain together and they get connected. And so now this is a functional mRNA. This is the one which is going to be used now to produce proteins. So this is what happens. Now let's go on and consider what happens during the process of regulation of gene expression. Now, gene expression or the gene regulation includes a wide range of mechanisms. There are a lot of things that have to happen. So it is not a one process thing. It is not a one-way thing. It has got a lot of mechanisms. So these are mechanisms which are used by the cells to either increase or decrease the production of a specific gene product. Okay. So a mechanism used by the cell to increase or decrease the production of a gene product. So this is what we are referring to as regulation, a mechanism used by the cell to either increase or decrease the production of a gene product. So this gene product, we've said it can either be RNA or a protein, because when we have got DNA is going to be converted into RNA in the process called transcription. RNA will now be converted into proteins in a process called translation. And so the conversion of these to any of these two products is what we call, so any of these two are called the gene products. Alright. Now, often the gene regulator controls another, and so on, and the gene regulator. So what is going to happen is you have a gene regulator which is going to control another gene, and it continues to go like that, and so on and so forth. So in eukaryotic genes, expression is regulated during transcription. That's the very time when we are converting DNA into RNA. That is when the regulation happens. And it also happens during the process of RNA processing, because when we form RNA from DNA, the RNA that is formed is called pre-mRNA. And so this has to undergo some processing so that it becomes mature mRNA. And so during that process, we are able to regulate how much gene product we want to form. How much should we increase the speed or reduce the speed? Alright, so this typically takes place in the nucleus during protein in the nucleus, because the conversion of DNA into RNA, DNA is found in the nucleus, and so even the conversion of DNA into RNA takes place in the nucleus, and so this process of regulation happens in the nucleus. Of course, regulation can also happen during the process of translation, when we are forming proteins from mRNA, and the formation of proteins from mRNA happens in the cytoplasm, and so it can also happen in the cytoplasm. Further, regulation of gene expression can also occur during post-translational modifications, because when we form proteins from mRNA, we have now to modify, because when we just convert mRNA into proteins, what we formed first is a polypeptide. So this polypeptide has to be modified. It has to undergo modification, and so when we do modify it, then it is going to be functional. So just the very time when we are modifying a polypeptide into a functional protein, we can regulate how much we want to form. Okay, we can control how much we want to form. So one common method of regulating the expression of eukaryotic genes is to alter the rate of transcription of those genes. We are saying one way we can be able to regulate the expression of eukaryotic genes. So here we are interested in eukaryotic cells, such as plant and animal cells. So one way we can be able to regulate the expression of genes, how we are forming genes, the way we can control the rate at which genes are formed, is actually to change the rate of transcription of those genes. Okay, we are going to change the rate of transcription. We know transcription is the process by which DNA is converted into RNA, so we are going to change the way this process is going to occur, the process of DNA into RNA. Now let's look at some things and see how that happens. So I'm going to draw one segment. Okay, let's put also the TATA, that is the data. Let's have another thing here. We are going to see their names and what they do. Now we can have introns and exons. Alright, so we can now label these. The ones which we can see in red, these are introns. And then the ones we can see in blue, these are exons. We have got this one which I have just put after the data. This is called the start site. Start site. And then we have got the data here, which is also called the core promoter. So these two together, they give us what is called the promoter. So I've got a first promoter and then the co-promoter. That means they work together. We are going to see the function. And then we have got an enhancer here. Well, during the process of transcription, we are saying one common way of regulating the expression of gene or of eukaryotic gene, if we want to control the way genes in a eukaryotic cell are regulated, if we want to control the way genes in a eukaryotic cell are expressed, we are going to alter the role of transcription. Instead of letting the process of transcription just happen like that, we are going to change it. And we know that transcription changes DNA into mRNA. And so it is mRNA which contains introns and exons. It is actually after mRNA has been formed, the introns have to be removed and then the exons have to be joined together, so that after then what we have formed is a functional mRNA molecule. So we have got components. So this is what we are calling a eukaryotic gene. So components of a eukaryotic gene. So when you are talking about a eukaryotic gene, of course we have already seen what a gene is. It's simply a segment of a chromosome that codes for a protein. So we are now talking about components of a eukaryotic gene. Just these things we are able to see here. So the first one that we want to talk about is an intron. Now, what is an intron? Introns are found on a eukaryotic gene. Well, introns, these are DNA sequences that do not code for anything useful. These don't code for any polypeptide, and so that is the reason as to why when you form mRNA, these introns have to be removed. So they do not code for anything. So we are saying they don't code for any polypeptide. And then the others we can see they are exons. And exons, these are DNA sequences that encode for polypeptide. So these, they code for a polypeptide. These are the ones which are going to be joined together after mRNA has been produced, and then they're going to give us a polypeptide. And it is from there that we even get a protein. Well, the other thing that we can see is a transcript start site. So we have got the transcript start site, which we can see there. Now, the function of this start site, this is the location along the DNA where RNA polymerase 2 binds to RNA polymer So this point, for the process of we want to convert DNA into RNA, this is the first point where RNA polymerase has to attach, because when you are forming when we are process of transcription has to occur, we have to convert this into this. And what does that? What converts DNA into RNA is the RNA polymerase. So that is what is going to happen. This is where RNA polymerase has to attach. And that is actually RNA polymerase 2. And so the next thing that we have there is a promoter. That's the fourth thing, the promoter. Well, as you can see, when it comes to the promoter, we have got two. We have got this and we have got the co-promoter. So let's start with the co-promoter. What is the function of the co-promoter? Now, the co-promoter is a sequence of DNA that is located about 40 bases, okay, upstream from the start point. So it is about 40 bases from the point where transcription has to start, from this point. So there are about 40 nucleotides. All right, so there are about 40 nucleotides. Now it is found in all eukaryotic genes. The co-promoter in whatsoever eukaryotic gene we are going to talk about, it is always found there. The most common example is the Tata box, which we can see, which I've shown there, the Tata box. That is an example of a co-promoter. Of course, we have got other co-promoters. Now, over 50 proteins can bind to the co-promoter, forming an important complex needed for gene expression. Over 50 can bind to this. And so if that over 50 what? Over 50 proteins are able to bind that. So when the proteins bind to the co-promoter, they are going to form an important complex, and that complex is what is responsible for controlling how much gene product is going to be produced at the end. Okay, so this is responsible. The promoter is responsible for controlling how much gene product is produced. All right, so the next thing that we want to talk about is when we are just talking about the co-promoter. Just under the co-promoter, we have an example of another co-promoter is a transcription factor 2D. So transcription factor 2, this is a complex that consists of the Tata binding protein. So transcription factor 2D consists of the Tata binding protein, which recognizes and binds to the Tata box. So for us to bind to the Tata box, this is a Tata box. We are saying a lot of proteins have to combine. Over 50 proteins can be able to combine to the Tata box and form an important complex that is responsible for controlling or regulating the gene expression. Now, what helps? Okay, what helps in the recognition of what is going to bind to the Tata box is actually the Tata binding protein. The Tata binding protein, it is contained by the transcription factor 2D. So transcription factor 2D is a complex which consists of the Tata binding protein, which recognizes and binds to the Tata box. Alright. Now another thing that we have on the co-promoter is the transcription factor 2B. Well, the transcription factor 2B, this protein helps the the Tata binding protein to interact with RNA polymerase. So that is the function. Because we know that RNA polymerase has to move along this strand in order for us to form products. So for RNA polymerase to move, transcription factor 2B contains, actually it helps TBT, that is the Tata binding protein. Okay, it is going to help the Tata binding protein to interact with the RNA polymerase 2. Let me write that. Helps the Tata binding protein interact with RNA polymerase 2. Alright, so that is the function. Now, apart from the co-promoter, we can see we have got also this one that is called the upstream promoter. Let's consider its function then. Well, the upstream promoter, that would be the second part of the promoter. The first one is the co-promoter, and then this is the upstream promoter. For the upstream promoter, this one, there are segments of DNA which are found upstream of the co-promoter. So they are found upstream of the co-promoter after the data box. So they can also bind activator or replacer proteins needed to regulate gene expression. So these can bind either replacers or activators, and this is very important when it comes to gene expression: an activator or a replacer. So a replacer is going to refuse the formation of a gene product. An activator is going to call for the production of a gene product. So these are two important things which are controlled by the upstream promoter. So the upstream promoter can bind or can bind the activator or the repressor which are needed to regulate gene expression. So unlike the co-promoter, upstream promoter vary from gene to gene. So the co-promoter is the same in all genes, but the upstream promoter is different depending on the gene that we are talking about. Alright. Now the other thing that you are able to see here is the enhancer. So what would be the function of the enhancer then? So enhancers are segments of DNA that are typically located far away from the gene. We can see where the genes are. The genes are very far, and the enhancer is just far away from there. Alright. Now, well, so these, they can be upstream or downstream. You can either find them this side or you can find them at the far end this side. Upstream or downstream. Now, enhancers bind special transcription factor proteins that increase the rate of transcription. These are going to bind what is called transcription factor, TF. So when they bind the transcription factor, this is able to increase the rate of transcription. That is very important. So when they do that, once the transcription protein binds to the enhancer, or the transcription factor binds to the enhancer can now loop around and bind onto the promoter region, thereby stimulating transcription of that gene. So what is happening is this: we have got a gene like that. It goes like this. So we have got a promoter. So when on the promoter, I mean on the enhancer here, not a promoter. So when on the enhancer, a transcription factor is held there. That is transcription factor. It is going to loop. It was like this, but it goes down like that. It loops. The looping helps is to bind to the promoter. So the promoter region is here. So this is the promoter region. So when it does bind to the promoter region, that is going to stimulate transcription of that gene. That is now going to say, okay, it is now time to start making a gene. So this is the importance. So all of these parts have got a function that they have to play. So after the transcription of the gene starts, the transcription factor as attached to the enhancer. The enhancer has now curved like that. It is now attached to the promoter. Polymerase will continue moving like that in the forward direction, forming in exons and introns. So those are now the transcripts or the gene products which will be formed. So this is what happens then. So we've seen how they are controlling. So each of these, they control the process. How fast or slow it moves depends with all these parts. Now, what is really the importance of the regulation? Why do we need to regulate the expression of genes? One important reason is the importance. We need to regulate the gene expression because it conserves energy. So it conserves energy. Not only that, also space is conserved. Imagine you are just producing things that your body doesn't want to use, which are not for use at that particular time. So that would be useless, right? That would be even taking up space in the cells. And so you are going to, because whenever you are doing this, you are using up ATP, you are using up energy. And so to conserve energy, you only produce what you're supposed to produce when it is necessary. That is to regulate. That is why we are regulating. You only produce what is necessary at a period of time. Okay. So another thing is the regulation of gene expression is important in that gene expression is regulated to ensure that the correct protein are made when and where they are needed. So that is it. Because we don't need to produce proteins every time and then. We only need to produce proteins when they are needed. And so that's why we need to regulate. We need to control how genes are expressed, how genes form gene products. So these are two important reasons that we need to get to understand. Well, in this video, end here. Stay blessed.